Assalamu alaikum guys. So moving on to prokaryotic cell structure. So prokaryotic cells don't have a nucleus, nor do they have any membrane-bound organelles. What they have a part of the cytoplasm that has a chromosome, one chromosome, and this is called the nucleoid. Okay, this part, this pink part is the nucleoid. It's a primitive nucleus, but as you can see, it don't have any nuclear membrane, and the chromosome is made of only DNA without any proteins. They do have extra chromosomal DNA in the form of plasmid, this yellow structure. And in the cytoplasm, they have ribosomes, they have inclusions, these are storage materials, and they might have spores or endospores. We will talk about each structure individually. Surrounding the cytoplasm, they have three layers combined are called the bacterial envelope. Okay, so the bacterial envelope consists of cell membrane, cell wall, and sometimes they have a third layer, which is the capsule or the glycocalyx. Surrounding the bacterial envelope, they have external structures like flagella, pili, and fimbria. So this is what we have just said. And the cytoplasm is the semi-fluid, thick, aqueous, transparent, elastic material that fills prokaryotic cells. The semi-fluid part, as in eukaryotes, also is the cytosol. The cytosol consists of 80% water, dissolved enzymes, oxygen, essential nutrients, proteins, lipids, carbohydrates, waste products, and inorganic ions. The bacteria has all these things in the cytosol, in the cytoplasm, because bacterial metabolism occur only in the cytoplasm. They don't have membrane-bound organelles like the mitochondria and other organelles. Moving on to nucleoid. The nucleoid, as we said, is the primitive nucleus, is the control center of the bacteria. A nucleoid is a part of the cytoplasm that contains a single chromosome of the bacteria. The chromosome is a supercoiled, circular, double-stranded DNA without any proteins. This is versus the eukaryotes. The chromosome contains usually around 3,000 genes, but this number can vary greatly. In comparison, the human genome contains from 20 to 25,000 genes. The plasmids are extrachromosomal DNA. They are small, circular, double-stranded DNA outside the chromosome and outside the nucleoid. They are not always present, and they are not necessary for the survival. They are the bacteria can function without them, function normally without them. If they are present, there can be multiple copies of the same plasmid or multiple different plasmids. Each plasmid contains from 5 to 100 genes. These genes, as we said, they are additional, but they are very important in certain situations. For example, the bacteria, okay, they can produce proteins that pump the antibiotic out of the bacteria. So this is the antibiotic resistance. Other way for resisting the antibiotics, the plasmids can code for proteins that degrade the antibiotic. Okay, so also plasmids can produce toxins, and it can produce enzymes. This is the comparison between the chromosome and the plasmid. Both have circular double-stranded DNA. The chromosome have around 3,000 genes, while the plasmid have around 5 to 100 genes. This is a single copy of the chromosome per cell, while the plasmid can be up to 20 copies per cell. Okay, so the ribosomes are the site of protein synthesis. The prokaryotic ribosomes are 70S in density, so they are smaller, less dense, and contain fewer ribosomal proteins and ribosomal RNAs than the 80S ribosomes of the eukaryotes. As in eukaryotes, the prokaryotic ribosomes are composed of two subunits, but these subunits are different. The small subunit of 30S and the large subunit is of 50S. And some antibiotics work by targeting these subunits, inhibiting protein synthesis in the bacteria and paralyzing the bacteria. Now moving on to the inclusions or the granules. As we said, these are stored materials. They are reserved deposits, and they have multiple types. So the metachromatic granules or the volutin, these contain phosphate. The sulfur iron granules contain sulfur or iron. The polysaccharide or starch granules contain polysaccharide, and this is used for energy. The lipid granules contain lipids, and this is used for energy. And lastly, the carboxysomes. These are very important inclusions. They convert CO2 into organic material, just like the photosynthesis, but without the need for oxygen. This is called carbon fixation, and they use ribulose 1,5-diphosphate carboxylase enzyme. The spores or the endospores, these are thick protein coats inside cytoplasm that contain a copy of bacterial chromosome. This is formed when the bacteria is dry or the essential nutrients are depleted. So when the bacteria is in danger, the environment is not suitable for growing, the environment is dry or the essential nutrients are depleted, some bacteria has the ability to form a coat around a copy of the chromosome, and this coat is resistant to desiccation, dehydration, heat, chemicals, or radiation. So in other words, we can't kill these spores easily. Okay, so spores can be central, subterminal, or terminal. Let's look at this slide. So this is the terminal spores of Clostridium tetani bacteria. The Clostridium tetani is the cause of tetanus. And as you can see, we have the circular spore inside the bacteria, and this can resist everything for the exception of pressure. So the sporulation is the formation of spores, and germination is the return to the vegetative state. The vegetative state is the growing state of the bacteria. As we said, only some types of bacteria are spore formers. These are two types, the bacillus and the Clostridium. The bacillus causes anthrax and causes food poisoning. The Clostridium causes tetanus, botulism, gas gangrene, and food poisoning. Both of these bacteria are gram-positive and rod-shaped bacteria.